Well, hello and welcome to Norwalk Community College Bio 212, Chapter 28, the reproductive system. And we're going to start the chapter by bringing you all the way to the end of the chapter. And that's because they don't address why it is there's a male or female or male reproductive system, the way that they're actually physically different from one another. They just leap right in and say, look, here's the male reproductive system, and oh, look, here's the female reproductive system, and look, here are some hormones that might be made and how you regulate it. I think when you're reading this chapter, you should always be thinking about the fact that, there are 23 pair of chromosomes, right? So let's put this thing up here like that. Let's go like that. There are 23 pair of chromosomes. No, P-A-I-R, chromosomes, and C-H-R. And two of them are sex chromosomes. Chromosome. Okay. And in those sex chromosomes, what you actually have on the male chromosome, the male Y chromosome, okay, let's go bold, let's go underline, let's italicize to make sure you understand the significance of this, is there's a gene, and that gene is called the SRY gene, okay? And what happens is, when expressed, a population of cells, sustenticular cells, or nurse cells, or Sertoli cells, okay, are going to secrete this molecule MIS. So what this means is your sustenticular cells, which are part of the male reproductive system, are in fact an endocrine gland, and they secrete this molecule called MIS, Mullerian inhibiting hormone or substance, right? And that's what's going to cause the system to physically change its structure and location such that you're going to drive the production of HCG, which is going to make in utero a small amount of testosterone in the shape of DHT, dihydroxytestosterone, and that's going to cause the actual structure and function development of the male reproductive system. And this happens beginning in utero, right? And so we are talking in the gestational period before birth, okay? And the important thing to hear is C as well, is in the absence of the SRY gene, which would be two X chromosomes, right? You would actually have the default pathway, which is the female reproductive system, okay? So that's why, I suppose it helps if I spell chromosome properly here, right? Okay. So that's how we're going to start chapter 28. Mother Nature, long before we need to understand what happens at fertilization, has to plan for the development of a series of ductal structures inside the male reproductive system that will allow for the passage of meiotic cells, and those haploid cells are called spermatocytes, which will have to travel into the female reproductive system, such that fertilization of the meiotic secondary oocyte will occur. Now that's a lot of talking. And I'm hoping you've actually read chapter 28. If you haven't, this is a really good way to start the chapter, so it sort of makes a little bit more sense while you're reading it. But at the end of the day, okay, it's expected that you'll read the book, you will watch these 3 or 4 videos, depending on how much time it takes. I try to keep everything underneath 20 minutes, if at all possible. But you'll have the details to make sure you can answer questions. Okay, having told you that, your textbook leaps right into the male reproductive system. So that's what we're going to do. The male reproductive system is made up of a series of structures that are called gonads, and these are testes, and they're going to make spermatocytes, collectively known here as sperm, as well as hormones. The major hormones we'll cover in a few minutes, but you already know them. You know them, or at least you know the major one, which is testosterone. Having said that, you should remember that testosterone is a steroid hormone. It is made from what precursor molecule? Yes, if you said cholesterol, that's completely correct. And like I said a moment ago, you actually have to get these materials, namely the spermatocytes, through a series of ducts where production will occur, transport and store sperm, as well as assist in their maturation, and will convey them to the outside of the actual body itself. They're going to have specific names: epididymis, ductus deferens, ejaculatory duct, and urethra. And as we're going along, we're going to introduce the fact that it isn't just spermatocytes that actually come out of the male reproductive system. There'll be accessory sex glands here that are going to be making materials from the seminal vesicles, the prostate, and the bulbourethral gland to ensure that material actually gets out of these ductal systems. Okay? How is it you actually have supporting structures? Well, we'll talk about the scrotum in a few minutes, and then actually how the penis is required to deliver sperm into the female reproductive system. Okay, so let's take a look at a hemidissected male here, deceased, and you can see that you actually have a bladder up here. And what you can't make out in this, hopefully when you're looking inside your textbook, is that you actually have a ductal system that starts down inside the testes. It travels up. I'm going to move up here to the cartoon so you actually see it. You can see it travels anterior to the pubic symphysis. It travels around, so laterally around the bladder, and then posteriorly behind this structure here. And you're going to learn that that structure eventually is going to be called the ureter. Okay, and what's going to happen is it's going to travel behind here. We're going to start to see some of those glands. Here's a seminal vesicle. Materials are collected from the seminal vesicle. Materials are made inside the prostate gland. They're mixed now with the spermatocytes that have been made here, and they travel with appropriate stimulation out of the urethra. Materials from the bulbourethral gland are now going to be present as well and allow for removal of the spermatocytes from the male reproductive system. So let's start down at that scrotum. Okay? When looking at the next image inside your textbook, you're going to see that the scrotum is actually the supporting structure for the testes. Remember I told you that in utero, in the presence of the SRY gene, okay, presence SRY gene, MIS is made. I'm going to give you MIS, and that MIS is going to allow for the actual rearrangement of the male reproductive system so that you actually have the dropping of the testes outside of the male reproductive system. The middle of the scrotum on the exterior surface, okay, is the median ridge. And you actually have the scrotal septum that actually divides the two testicles into right and left halves. And there's made up of a subcutaneous layer as well as a dartos muscle. That dartos muscle will become a little bit more important in a moment, as well as the cremaster muscle. Okay. The reason why your body actually keeps the testes outside is that they need to be about 2 to 3 degrees below core temperature, and the cremaster muscle and the dartos muscle work together to ensure that the appropriate distance for the testes away from the body will allow for proper development of the actual spermatocytes. You can see this here. Here's that raphe we were talking about a moment ago. This is the external scrotal sac. You can see here the dartos muscle, which is deep to the skin of the scrotum. And as you move inward, you can see the structure that's called the tunica vaginalis. It surrounds the actual testes itself. And you're going to have this outcropping of ductal structures. It's called the epididymis. Materials made inside the actual testes here travel through that spermatic cord, otherwise known here as the vas deferens or ductus deferens, and then will travel around the side of the bladder, as I indicated earlier. This is a transverse section of the penis, and what I want you to see here before we move on is you actually have 2 cavernosa. Okay, and what I mean by that is you actually have 2 corporal structures. This is the corpora cavernosa of the penis, where what happens is you're going to fill these areas with blood. Simultaneously, the corpora spongiosum will also be contained with blood or fill with blood. And this is what's actually going to change the shape of the actual penis upon arousal. Okay, so I told you a moment ago that they're paired ovular glands, and I never said that they were ovular, but it doesn't really matter. Okay, they're developed near the kidney, and it descends through the inguinal canals at that 7 month. Okay, roughly. Okay, and this is where we actually put in the MIS again, because without the MIS, what wouldn't you develop? You wouldn't develop an external ductal system. Okay, I mentioned to you the tunica vaginalis, which partly covers the testes itself. And what I want you to see when we actually cut through this in a second, you're going to see that each lobule inside of the testes itself has about 200 to 300 subsections, each containing 1 to 3 seminiferous tubules. And that's where the important process of spermatogenesis is actually going to occur. Okay, so I haven't been doing a lot of drawing yet. That's okay. We're going to start introducing some concepts in a couple of minutes here. And what I want you to do is get used to the idea that you're now looking at a process. You're looking at a process where materials are going to be made in location A. A series of signals will regulate that production. And then we have to figure out how we get materials out of the body. Okay. So here is the one of the testes. Okay. And what they're doing here is they're putting an imaginary sagittal plane through the cartoon for you. Right. So let's go like this. Let's go like this. Let's go like that. Let's go like that. Okay, and what I want you to see is that imaginary plane literally is boxed here. And a moment ago I told you you actually have these lobules. What I want you to see, maybe you'll actually see this inside lab when you actually do your dissections, you're going to see that these lobules are deep to that tunica albuginea, and you see these little septa that separate what are going to be called the seminiferous tubules. Okay, so inside of each of these lobules, you have seminiferous tubules, and this is where spermatogenesis is going to occur. What I need you to see here, and what hopefully you're paying attention, close enough attention to pay attention to see, is that if spermatogenesis occurs here, then, I probably should have picked a better color here, you're going to have movement of materials into the rete testis. The rete testis is now going to move materials up into the epididymis, where inside the efferent duct, maturation of those materials will occur. And you're going to notice that these are all really convoluted, right? And that means you have more time spent here. And as it's maturing here, it will eventually travel out through that ductus deferens and then out of the spermatic cord. And even though this cartoon probably only has some blood vessels and some nerves inside of it, this has to be richly vascularized. Otherwise, there's no glucose, there's no oxygen, there's no removal of carbon dioxide, and therefore metabolism couldn't happen. Okay? So let's get rid of that. Let's do that. Let's do this. Let's move on to the next slide. So what's actually going on inside of here? There's a lot. Okay? And I'm not going to read the entire slide to you. You're an adult. You can read. What I want you to do is see that I actually have a series of arrows here that are going to help you figure out that we're going to move in spermatogenesis from a developmental cell. Okay? And remember from AMP1, anytime you see a gonia attached to something, it is a primordial stem cell structure. Okay? And it's going to roughly, okay, with time, with sufficient stimulus, okay, are going to ensure that this primary spermatocyte becomes a secondary spermatocyte, which will ultimately become a sperm cell, which to leave the body has to occupy the lumen of that ductal structure. How does Mother Nature make sure this happens? Well, what she does is she takes a series of cells. Depending on which textbook you're using, you could be using Tortora and Derrickson. For all I know, you're using something else. Okay? You have Sertoli cells are sustenticular cells, are nurse cells, which are support cells. What happens is they form a tight junction to create this blood-testis barrier. Because I need you to think about something. And that something is, if we're going to undergo meiosis to make individual spermatocytes, it means that the immune system is going to see those sperm cells as other. Okay? Which are now other. I'm going to just put that. And what I mean by other is they have their own unique antigens now on the surface of their cells, and therefore your immune system would see them as foe and try to destroy them. They're also responsible for making sure that you actually have materials. They're going to make a specific type of hormone. It's called inhibin, which is going to regulate the effects of testosterone and follicle-stimulating hormone. You remember follicle-stimulating hormone from chapter 18. If I asked you right now or put you at the board and said, explain to me how we made follicle-stimulating hormone, you should be able to tell me the entire pathway. It's the Leydig cells. These are also called interstitial cells, depending on your textbook. Okay, they're going to be the target of luteinizing hormone, right, which is now going to make testosterone. We'll see how in a few minutes. Okay, cool. Spermatogenesis. Anywhere between 65 and 75 days. It begins with a spermatogonia, which is a diploid structure. I'm oftentimes surprised at how much I have to go back to fix people's understanding of this, but this means you actually have two pair of each of the chromosomes, one from mom and one from dad, and you have primary spermatocytes, which are also diploid. Okay, it's going to undergo a mitotic, the spermatogonia undergoes a mitotic-like event, okay, to replace themselves. A subset will then become the primary spermatocyte, which duplicates its own DNA, and that's where meiosis will begin. So it's the primary spermatocyte that actually will have meiosis beginning. Meiosis 1 is when we actually take homologous pairs now of duplicated DNA, and we literally, intentionally excise and exchange genetic information so that you actually have what's called a recombination of that genetic information, so that you end up now with essentially new chromosomes that you didn't have with the spermatogonia. Okay? So it's going to, by the time it finishes, you have two cells at the end of meiosis 1, and you have each chromosome, which is made up of two chromatids attached to a single centromere. Now we actually have to reduce this. That's the tricky part. We need to make 4 haploid cells from these now up here diploid structures, which will become secondary spermatocytes, which are haploid, and now we end up with 4 haploid cells. I'll go through the cartoon inside your textbook in a moment. Okay? They remain attached. They have a cytoplasmic bridge. Mother Nature wants to ensure that there's an equal amount of cytoplasm between each of these. So even though they may be genetically distinct, they have essentially the same starting material at the racing line, meaning when everything's all lined up, ejaculation occurs, everybody should have the same mechanical properties and enough energy to get to where they need to get to. Together, once you actually have spermatids in place, we now have to have them mature into sperm themselves. And the spherical spermatids become elongated. The acrosome and flagella will form as a result of this. And the mitochondria, okay, the mitochondria here, these didn't come from mom yet. The mitochondria here are multiplying. And that's because you learned back in AMP1, when you need more ATP, you can make more mitochondria. Sertoli cells will dispose of excess cytoplasm. Spermiation, which is going to be when you actually release the connections up to the Sertoli cells, and they're not able yet to swim here. Okay? What's going to happen, this is the cartoon where spermatogonium will undergo that first meiotic type event where a subset will stay behind, but one of those two will be committed to the differentiation pathway, where the first phase of meiosis allows for that DNA replication, tetrad formation, and crossing over. This is the recombination event I was telling you about. The products of this, the spermatocytes, now undergo meiosis 2, where you actually end up taking these... Whoops, I don't know what happened there. Sorry about that. This happens every once in a while. One of these days I'll figure it all out. You're going to take these and you're going to make now 4 haploid cells, okay? And the spermiogenesis is occurring now deep into the moving into the lumen of the seminiferous tubule here, such that you can actually see the formation of the flagellar tails. The mitochondria are multiplying, so on and so forth. By the time you actually have sperm that are now maturing inside the ductal structures of the testes, you have about 300 million that complete spermatogenesis per day. It has 23 chromosomes. Remember, they are haploid or a single N. The acrosome is filled with enzymes that can actually break down the oocyte itself, and specifically, you put secondary oocyte here. And, whoops. The actual sperm itself has a neck, which is where you actually have microtubules, which are going to make the flagellar tail. Middle piece, which is going to be mitochondria. The principal piece, as well as the terminal piece. This tail is just moving around. The sperm do not survive for more than 48 hours inside the female reproductive tract. I'm sure that, 1 or 2 could survive a little bit longer than that, but on the whole, it needs energy and there's only so much that's available in the female reproductive tract. Okay, so this is a quick cartoon. Notice it looks nothing like all the other cartoons you've seen before. Okay, and what I want to do when I'm finishing this up is I want to introduce this idea that now at 20 minutes, we have a series of hormonal controls, and that's where we'll pick up in the next video. Okay, so again, remember the process to be successful inside this class is you read the textbook first, then you watch these, and hopefully you're going to figure it all out from there. Okay? Inside lecture, obviously I'm going to help you focus on that.